back I got.
Which one, no?
I got Nissan gun. You not seen?
No.
Long back I got, almost 2 years.
2 years.
That is also there. Both are there.
Wagner also is there, no?
Both
You
are.
didn't dispose it all.
That my son wants to use.
Oh, okay.
Both is using,
Ah.
but he's taking for college now.
Okay.
I got long back I got.
Ah, good there. Nice one.
Low night. You saw the game today.
I saw, but I thought maybe he came by cab or someone would have come.
He told my son attend. He told me 2 o'clock he has class. Here Anand is waiting from 8:15. He's calling me. I told I'll come back after. No cab is already declining.
Where you picked him up?
He comes to MG Road, from there we come. So he was sitting calling, where are they? Sir, I'm coming by own car, I have to come. Then I picked him up. He got, he joined college now, so his grandfather got him.
Okay.
So he sponsored people. Nowadays all are coming by car, from two-wheeler, no value there. So when you go in car, security will salute. Otherwise two-wheeler will chase him out. Don't tell car.
No, whom? I think I am the last one to hear about it. They would have been circulated already. Huh?
Maybe in Facebook they would have seen. What I don't bother now.
What is that? What is that?
I buy helicopter also. Who will tell? See a normal security guard. Muniappa, how nicely house is built.
Is it? Yeah, they told they had gone for
How
husband and
beautiful.
family.
1+3.
Is it? Oh my God.
Then where is our capacity car? We will be like this only forever if we don't improve. Correct, no?
ಚೆಕ್ 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 ಬರ್ತಿದ್ಯಾ ಸರ್ ನೀವು ನಾನು ಇರ್ತೀನಿ ಹೇಳಿ ನಮ್ಮ ಹುಡುಗ ಸ್ವಲ್ಪ ಹಿಂಗೆ ಇಟ್ಕೊಂತೀವಿ ಸರ್ ಬಿಡಿ ಸರ್ ಏನೋ ಮಾಡೋಣ ನಾವು ಮುಂದೆ ಹೇಳಿ ನಾವು ಮಾಡ್ತೀವಿ ಸರ್ ಬರ್ತಿದ್ದೆ ಆಟೋ ಚೆಕ್ ಮಾಡಿ ನೀವು ಸರ್ ಓಕೆ ನೋಡಿ ಚೆಕ್ 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 ಸಾಕಾ ಚೆಕ್ ಚೆಕ್ ಚೆಕ್
Check, check.
Very warm welcome to all of you. We have Professor Sir Tom Blundell with us. We have Madam Kiran Mazumdar with us. We have faculty from many different institutes, students, everybody else. It has been a very important day this year in IBAB, particularly because the prestigious Kiran Mazumdar lecture, which usually happens every year, could not be conducted during the past couple of years because of COVID pandemic. So I'm very happy that we were able to resume now, and this year we are going to have the lecture. Dr. Kiran Mazumdar endowed this prestigious lecture series with the objective that it will help us in bringing some of the most innovative and reputed scientists to IBAB in particular, and the Bangalore community in general. The lecture series has been running for the past over 10 years. The first lecture was held in 2011, and the lecture was given by Professor Venki Ramakrishnan, who is a Nobel laureate, as you well know. Since then, many highly reputed scientists have given the lecture as part of this series. They include Professor Dinsha Patel, Professor Ram Shashishekaran, Professor Sangeeta Bhatia, Professor Arul Chinian, Professor Ashok Venkataraman, and Professor Vamsi Mutha. Just to give a very brief introduction, although probably nobody needs it really, about Kiran. She is the executive chairperson of Biocon and a global influencer of and a passionate philanthropist. She is the pioneer of India's biotech industry, as you know, as founder of Biocon. She is the proud recipient of two of the India's highest civilian honors, the Padma Shri as well as Padma Bhushan. She has been conferred with the EY World Entrepreneur of the Year 2020 award, which is the testimony to her entrepreneurial journey over the decades. She has several other international accolades. I won't get into the details of it. As an example, the highest French distinction, the Knight of Legion of Honor in 2016 and Otmar Gold Award in 2014. She serves on the board of several prestigious institutes. As an example, the MIT and the PureTech Health, Memorial Sloan Kettering, Infosys, Narayana Health, and so on and so forth. Kiran, apart from all her accomplishments and honors and awards she has got, we are very fond of her because she is instrumental in establishing the Institute of Bioinformatics and Applied Biotechnology, IBAB. As a chairman of the Vision Group of Biotechnology, the institute was established on her recommendations, and since day one she has been nurturing the institute in every possible way. IBAB has reached the heights it has reached today because of her constant support and help. So thank you very much, Kiran, for all that. Now to introduce Professor Tom Blundell, or Professor Sir Tom Blundell. Again, Tom is known in scientific communities in India and in particular Bangalore for a very long period of time. I'm pretty sure that everybody knows about Professor Tom Blundell. He is currently the distinguished professor at the Heart and Lung Research Institute, the Department of Medicine, Royal Papworth Hospital. Earlier, he was the 5th Sir William Dunn Professor of Biochemistry and head of the Department of Biochemistry at the University of Cambridge. Professor Blundell actually started his career in the Department of Chemical Crystallography, first with Herbert Marcus Powell, for his PhD, and then later working on insulin with Dorothy Hodgkins. Over the years, he has been involved in or contributed to the structure determination of a large number of protein structures, such as aspartic proteinases, serine amyloid proteins, and stromalysine, and so on and so forth. More recently, I was very astonished, really, just a few years ago, they have determined the structure of a very huge protein, a 4,000 amino acid protein, the DNA PKCs, which was published in Science a few years ago, which helped in suggesting an allosteric mechanism of modulating the DNA double-strand break repair. His group is also known for significant contributions in the development of new structure-guided softwares, for example, the Composer and Modeler. I was just mentioning, Professor Bundle, that Modeler is a component of the curriculum at IBAB. We teach Modeler as part of it. Yeah. Just to give a popularity of the softwares which were developed by his group, if you take Modeler has more than 11,200 citations worldwide. So that is the use the computational biologists have found with the Modeler software. Apart from the academic career, he also co-founded a company called Astex Therapeutics in 1999. Besides his research activities, he has been contributing enormously to the national science policy. He was the Director General of the Agriculture and Food Research Council and the founding Chief Executive of Biotechnology and Biological Sciences Research Council, BBSRC in UK. He has been conferred with very prestigious honors such as the Fellowship of Royal Society, membership of the Academy of Medical Sciences, and to crown it a knighthood in 1997. Besides his academic accomplishments, as I mentioned earlier, Professor Blundell has extensive links and long-term collaboration with Indian scientists. It is very fortunate, sir, that you have agreed to deliver the 8th Kiran Mazumdar Shah lecture today. The title of the lecture is Structural Biology, Bioinformatics, and Drug Discovery, Targeting Cancer, Mycobacterium Tuberculosis, and COVID-19. Very warm welcome to you, sir. I leave the podium to you.
Well, thank you very much for that very kind introduction, and I should say it's a great honor to be giving the Kiran Mazumdar Shah lecture. I apologize that it's not on my slide because I made the slide several days ago and I didn't realize that it was part of a formal set, but I'm very honored that that is the case and we can celebrate the fantastic contributions that Kiran has made. Looking at what she's done, I'm just hugely impressed. I've tried to do the same thing myself on both policies, international activities, companies, and so on. And I'll say a little bit about that as I go along. I, of course, have been coming to India for many years, as was just mentioned. I was a co-worker with Vijayan, and Vijayan and I helped solve the insulin structure in 1969. So, what is it, 50-something years ago, we made that major breakthrough. And it was very important because insulin was involved, obviously, in treatment of diabetics. And they were actually treated using very tiny crystals so that they didn't have to be injected many times. So I started off in the environment with Dorothy Hodgkin, the Nobel laureate, whose husband was a member of the Communist Party. She had obviously been very tolerant of her husband, and when she found that I was running the city of Oxford as a left-wing counsellor for quite a few years. She was very, very understanding. So that's where I started. So I'm going to talk about structural biology, bioinformatics, and drug discovery. And I will divide it up a little bit. I'll talk, of course, about human genomes. They're absolutely essential to understand. And I've done a lot of computational work in trying to understand them. But we also need to know about, in my case, mycobacterial genomes. I've worked on tuberculosis, cystic fibrosis, which is a mycobacterial infection of the lungs, and also leprosy. And the International Leprosy Mission has put their central lab now in my site in Cambridge. I'm afraid that means they moved it from India, which was a bit of a shame. I felt rather guilty, but I'll say a bit more about that. And I, like everyone else, has spent a little time during the pandemic seeing whether the things that I've been doing about genomes and function could be extended to SARS-CoV-2. I recently, it's 60 years since I was doing my first structural biology, and that was with Dorothy Hodgkin and Tiny Powell, both members of the Royal Society. But 60 years later, everybody accepts there's been a new revolution, and that's the revolution in cryo-EM, where we can image proteins. Now, I should say many of us have been doing that for many, many years, and there have been developments recently, not so much, but to some degree on the equipment, but also on the software. And so we're now in a position where we can image almost everything. And in my lab, like many others, they removed just about everything which was X-ray related. So I'll talk a little bit about this giant molecule, DNA-PK, which in some ways summarizes what you can do with structural techniques, including cryo-EM. So my talk won't just be historical. And then in parallel, the other revolution has been AI, ML. And I should say I've been using machine learning to write software for I think 13 or 14 years in a very focused way. But there have been really important developments in its use, and those two have really transformed everything. So I'll say a little bit about that. And I'll also talk about how I've used all that background of both computational and experimental techniques to found a drug company and have drugs on the market now, one selling over $1 billion. Finally, I will talk about emerging resistance. It's actually absolutely critical that we understand how resistance develops so that we can design new drugs which are less likely to exhibit resistance. And I've spent quite a lot of time on that area. And so that's just a little bit on machine learning. So it's going to, I'm afraid, to be a little bit historical. And when you get old, that's always a failure of people. I'll start with J.D. Bernal. And J.D. Bernal was, of course, an amazing individual who, in my view, was not politically recognized and didn't get the Nobel Prize, and I think he should have done. He was a left-wing person who recruited Dorothy Hodgkin, but also worked a lot on social function of science and science in history. And it was actually the J.D. Bernal books that influenced me when I was in my teens. I started reading them and wondering who this guy was. And then, to my surprise, I found that his main personal influence on proteins was Dorothy Hodgkin. And she was still in Oxford at the time. And so I very quickly did a PhD and then moved to her lab. Now, why did I very quickly do a PhD first? The answer is that she'd already been working on the structure of insulin since the 1930s, and this was 1964 when she got a Nobel Prize. So 30 years and she hadn't solved it. I thought what I would do would be get my PhD as quickly as possible, learn the techniques, and then I could spend time trying to solve this problem with other colleagues, of course, and that's what I did. But what happened is that Bernal and Dorothy Hodgkin, who went from Oxford to Cambridge for just a short while, realized that the reason why they couldn't get crystals, proteins to diffract using X-rays was that they were not sealing them off, and you have to capture them between water reservoirs, as you can see in that picture. Once they saw that, they got diffraction, and this was early on proteins like pepsin, and that was what they wrote in Nature. The paper went straight into Nature in a week, I think, and they said that now we can solve the structure of any protein if we can get it crystalline. So that was a very important event. And I joined the team of Dorothy Hodgkin much later, so in the 60s, but still quite a long time ago. You can see that it actually has people from different countries. I was one of two English persons in the group, but a very important person was Vijayan, and Vijayan and I spent as much time talking about politics as we did about science. He spent a year in politics before he came back to science, and was very left-wing, so that was a very interesting discovery. And the great thing about it, it was multinational, it was gender balanced, and it really was an amazing place. We had continuous parties and all celebrations, but we're not only celebrating ourselves having drinks, but also, of course, making some quite important breakthroughs. And that's a picture of me in 1969. And what happened in that case was that I was going to my first international congress where 3,000 people were there. And as I walked in the day before it started, the organizer rushed up because I was with Dorothy Hodgkin. He knew Dorothy. And he said, "The first speaker tomorrow, Dorothy, has cancelled. And could you give the talk? You just solved the structure of insulin." And Dorothy said, "Oh, that would be lovely." She said, "I'll say a few words and Tom will give the lecture." So I had to spend all the night making slides of things we've done in the last previous few weeks on insulin. And that worked rather well in the end, so I was very lucky. And that was my first exposure to big groups. But Dorothy Hodgkin, of course, have been working on the insulin crystals, and they're the ones that are used in very microcrystal form to treat diabetics, and that's because they dissolve rather slowly. And so we were able to use X-ray diffraction to get that beautiful structure of the protein, which we published in 1969. And that's my sitting, as I often did, talking with Dorothy about politics and science and all sorts of things. She was an amazing person. So that's how I started. But I realized that I was in Oxford and there was a problem. They were building a big road coming in from the west of Oxford, and it was going to go right through the centre. And all my everything in me said that would destroy Oxford, and I became very politically active. So as it developed, I stood for the city council. I was elected the first time, and then within 2 years further, I got a majority of people, because even the Tories agreed with what I was saying. So it was really party. Everybody agreed they didn't want a motorway going through the centre. And so I took control of the city and stopped the motorway. I pedestrianised the centre. I made North Oxford an area where it wasn't listed buildings, but it was a sort of conservation area. So that was my experience in politics early on. And so that's where the road was going to go. That's some of the beautiful buildings in the centre that no longer were juxtaposed with a major road going through. And that's one of the houses in North Oxford. So then I thought, what am I going to do? Am I going to be a politician? Or am I going to be a scientist? Or should I be a musician? And I said to Dorothy that what did she think? And she said, Tom, I think you better go away and think about it. So I don't know how I got the money, but I went on the train through Europe up to Leningrad. These are the things, by the way, that I did in Oxford, but I just mentioned those. So I went on the train up to Leningrad, and then I took the train right across the Trans-Siberian. I got stuck on the border of Khabarovsk, and there was no way of getting across. I thought I'd be able to hop across to Japan. But I was very lucky, because suddenly, as I was waiting, wondering how I can get out of Khabarovsk and go to Japan, a beautiful young lady and a very old man arrived. And I wondered what they were doing. And then I realized that, they said that there was, of course, a battle between the Japanese and the Russians in around 1930. So this was 40 years later. And the Russians were inviting the Japanese to try and make friends and build up a new sort of consolidated Eastern structure. So I've then realized that this lady had an old man she was taken back to Japan, and a plane with lots of empty seats. So I managed to persuade her that I get on it, and then I ended up in Japan. So I spent quite a lot of time in Japan learning Japanese, as I said. Nihongo benkyou shimashita. Not that good now, I was better. And then eventually I moved on and along the coast through Taipei and Hong Kong, really looking at the politics, the music and the science as I went. And this is, for example, a little bit later actually, but me giving a lecture in Beijing just after this time. I went back, they invited me, and I got these pictures. And you can see I'm giving a lecture, and above is a picture of Mao. So that was very interesting and exciting. Dorothy Hodgkin had been there a lot and knew everybody, it seemed. So, and then what happened later is they found out that I was a sort of jazz musician, and they made me play. So you can see, having given a talk and everything, I ended up playing the guitar for them. So, by the way, I'm not going to do that for you. I was much better in those days. So then I continued on, and I ended in Bangalore. And that's why I thought it was relevant to give you this little bit of history. And in Bangalore, I, of course, met Vijayan, and I got very much involved in things. But I went from Bangalore down to Karnataka area and bought a Veena. So I went all the way back with the Veena. But I'll say a little bit about that. So this was Dorothy Hodgkin in Bangalore in the 70s. That's Vijayan and myself in a train in Bangalore. And and here's a picture of the Vina, which I began to play after that journey. That's not me playing it, of course. And then I went back and wondered what I was going to do. And Dorothy Hodgkin said, you've been much too involved in politics. What you ought to do is just focus on science for a while, and you better go away and leave Oxford. So I said to Dorothy, but where shall I go? She said, come back tomorrow and I'll tell you where. So I went back the next day and she got me a professorship in Sussex. It's totally immoral, really. And so I moved to Sussex, and then I gave up my politics, and I did some work on the structure function of insulin, and then I solved the structure of glucagon, which of course is the yin and yang of blood sugar control, and that went into Nature as well. And then I started thinking about evolution, and I wrote a paper about selective mutation analysis and selectively neutral mutations, which hadn't really been analysed in terms of structure and function, although people were aware of it. So that's a paper in, I think it was in Nature as well, wasn't it? Yes, in 1975. And then in my spare time, to my surprise, I wrote a book on protein crystallography, because I realized that that has 500 pages in it. I don't know, I get up at 5 o'clock, by the way, and work very long hours. And I realized that the previous 20 years of analysis of proteins had not been written down anywhere. And so I wrote a book with 500 pages with Louise Johnson, who was actually at a similar stage in her career, but working with David Phillips, who came to Oxford. So that was all great fun. And then, to my surprise, I got offered a job, and that was in London. And of course it was near the Opera House, which was a main attraction in Birkbeck College. It was near the Jazz Club, but the main thing for me was it was actually the department that J.D. Bernal founded 20 years ago. So I became his, I guess, inherited his professorship. And that was amazing for me. I spent 20 years in London. I was only 34 when I got my professorship, and I had 150 people. And we published, I don't know how many Nature papers, about 20, at least one a year. And so it was very good. And the other thing is I got people from all over the world coming to join me. And we did all sorts of things. We thought about how we might do some drug discovery. We had these proteins. I mean, could we fill the pockets in a structure-guided drug design and keep the hydrogen bonds? And so the whole area of developing structure-guided medicine began to evolve in our lab. And I published a lot of this. I founded a company, but then Margaret Thatcher, to my surprise, right-wing Tory, asked me to advise her. And then I found I had to give up any collaboration with industry. So my first company collapsed under the pressure of what was proper. If you're a scientific advisor, you don't have companies that may cause a conflict of interest. Very depressing that was. But anyway, we did a lot of speculative work, and one of them was that I was looking at the aspartic proteinases and realized that structure on the left-hand side is actually evolved from something like the one on the right-hand side, which, in other words, a symmetrical dimer. So all the folding is actually conserved, although it wasn't obvious, and it took me some time to realize. So I then wrote a paper, again in Nature, predicting that the molecule on the right-hand side was the ancestor of these aspartic proteinases like pepsin and renin, which is involved in blood sugar control. And what happened is it took me 6 years of looking, and everybody, including Max Proust kept coming and telling me I was doing the wrong thing. He said, you're doing all this speculation, Blundell, why don't you do some proper work? But I found that molecule eventually. And where did I find it? I found it in HIV, and HIV is made as a single polypeptide, and it needs a peptidase to cleave it. So I suddenly realized if I inhibit that enzyme, I've stopped the HIV. And so suddenly we got a huge activity in making HIV medicines. And of course, other companies took it forward, but we certainly pioneered it, and it worked very nicely. So HIV is a polyprotein, and you just got one protein, which is the protease, and that clips it all up to individual proteins, and then the HIV is active. So we inhibited that. I found another colleague of mine was doing the same thing, and so we published in Nature back-to-back on that. And so new drugs in industry, but the other thing that really took my attention was the development of resistance, which was really growing quite strongly. So what did I do? I thought that I perhaps ought to think more about drug discovery in a systematic way, having made all these observations. So I realized that there were, well, everybody realized that there are two steps. First, you have to understand what the protein is like if you've got a gene. So can you predict the protein structure from the gene? And then secondly, can you predict what might be a new drug and bind to that protein? So I saw it as two separate parts, and we got working on those, and we could show this picture before, but we had the pockets of that molecule I've been working on previously, and we used the techniques to develop that. Yes, this is the renin and the pepsin. I seem to have some duplicate slides here. I apologize for that. But what we did is to follow at a molecular level all through those stages. I seem to have the slides in again. And we also began then to predict new drugs that would bind to the HIV protease, and in a structure-guided way, and that seemed to work well. But of course, you got resistance. And so I spent a lot of time trying to understand the nature of resistance and how we might avoid it. So that's what we were doing. But I'm going to jump a little bit to what's been happening in the last 30 years, really. And that is that we have a whole lot of techniques, small angle X-ray scattering, a lot of prediction software, and X-ray methods, which we're not using so much now, using nanospray mass spec, but of course cryo-EM. And the big resolution revolution in the past 6 years has obviously been the cryo-EM, and I was really impressed to see that there are at least several machines in this part of the world, and you were really up to date with using them. But almost all of my group has turned to defining these huge structures. This is the DNA PK with the 4,000 amino acids. I could get it using cryo-EM, and it went it went into a good journal, not into Nature. But then we also could look at complexes of this which occur, and these are complexes involving multiple proteins, and you could do that by cryo-EM. So what's happened in our lab and in most labs is that the X-ray machines have all been removed, and it's all filled up with cryo-EM facilities. I had a quick walk through your labs, and it looks like you were going in a similar direction, and that's, I think, sensible. So a lot of things happening, and you can see we have a lot of very talented people working in that. I've been very careful to keep a large number of women in the group, and this was done by Amanda Chaplin, who, like most people who do well in my group, then you go and get a professorship somewhere else, and I have to start again, but she's in the University of Leicester. But that's all very exciting. And so let me go on. So I spent a lot of time also thinking about how we can avoid the crystal structure work. And as you may know, and I think some of you have been using the software, I developed in my spare time a lot of software. There's two pieces of software, and I gather that Modeler is used quite widely here still. But that was something we wrote, as you can see, around 30 years ago. But that was using statistical methods. And then to do modeling by homology, you had to have some sequence structure alignment to get the sequence into the context of the three-dimensional structure. And so we did a lot of work like that and software developing. By the way, André Chany, who wrote that, is a famous structural biologist and member of the National Academy. He's based on the West Coast. And then what we did is to develop databases for things like Mycobacterium tuberculosis. So using the software that we'd use for modeling to model all the gene products. I was constantly redoing and doing structures to see whether they were realistic. And then we also worked on Mycobacterium abscessus. That's important because in cystic fibrosis patients, you need to really target that as it infects the lungs. And so that's been a big development. So we developed on all these cases. We can, using the software like Modeler and other more recent stuff, we can model about 80 to 90% of the proteins with reasonable accuracy. So we've done that for a lot of different things. And this is what I've been working on very much recently. It's leprosy. I have to admit, I think most people don't know what's happening in leprosy. And I didn't really understand, even though I'm spending a lot of time in India. But as you know, if you a member of the family gets leprosy, they'll almost always go immediately into isolation. And then, of course, the family has to make sure that they're okay. And I think they're very much challenged. So the American leprosy mission, and the main director of drug discovery is Sandeep Chaitanya, who's an Indian, but he's now working in my lab. We've been using all the techniques that we had on TB and other things to get new drugs for leprosy as well. And SARS-CoV-2, as I said, we've been doing a lot of work. I haven't time to go through, but we've been doing similar things. So this is the sort of thing we've been doing. I've been all through the COSMIC database, which is the cancer-related genes, and had a look at those and built models of them, and done quite a lot of work on predicting those. So that's all of what we've been doing there. But most of what we've been doing on drug discovery has been done in the company. And that's at the bottom there, Haran Jyoti, who I mentioned before. And we, together with Chris Abel, formed this company, Aztecs. And that was based on a new idea, well, one that hadn't been implemented. I guess a number of people were thinking on it. Is about using fragments. If you use a tiny fragment, just a fraction of the molecular weight of a drug, then you can get away with screening for 1,000 fragments. So once you've got a hit and you define the structure, you can then elaborate it and go to a drug-like size. And lots of people now are doing this, but we pioneered this in the late 90s and formed a company, Aztecs, using this, mainly working in cancer, but I worked on lots of other things in the lab. So this was Harren. We very quickly got 80 employees. I now got several hundred employees by the way in Cambridge, and we got a big building in the science park, and it's all been very exciting. And these are drugs in clinical trials, and this is a statement about the power of cancer research and drug discovery clusters in Cambridge, UK, has prompted Japanese pharma giant Otsuka to buy the science park medical technology pioneer Aztecs. So that was a statement saying that we had to sell the company, and we sold it to a Japanese company, and I think that's proved to be very sensible because they've weren't interested in just looking through and taking what's useful and closing everybody down. So I've still got everybody employed, which is my main worry. So that's all of the things. We've got the drugs in Aztecs, and we've got a lot of work on neglected diseases as well. I'm not going to go through this very much, but we've spent a lot of time with Bill Gates using the fragment-based methods, and he put a lot of money into our lab and been extremely helpful. So we've got a lot of work going on tuberculosis and that was very important because the lady in the middle there is my wife, who's Black African, Matabeleland, but she came to England 50 years ago, and they're my two daughters. I sent them all to local schools. One got a top first in Oxford and another a top first in London School of Economics. So they've done extremely well. But there are their nephews still in Matabeleland, much more difficult to get education in the same way, but I'm sure that they're just as intelligent. So we've done lots of designing new methods with fragment-based discovery. I'm running out of time, so I'll just whip over that. So we've been going through, for each of the mycobacterium, screening using fragments and elaborating them and getting hits. So that's a lot of fragments bound to a protein, and then you elaborate it and get a drug-like molecule from it. And then what happened is Bill Gates wanted to come and see what we were doing with his money, so he turned up on his own, and so that's him walking with my co-founder of Aztecs up in the lab. He insisted that I didn't tell anybody else in the university he was coming. He wanted to come and talk with us. We had amazing discussions. It was fantastic. And so I made him put a lab coat on, and then I put my students in front of him. I sat at the side, and he interrogated the students. I think they did pretty well, actually, on what they were doing, and it was all great fun. So we got new drugs, and everything's going well. Now, there's just been a revolution, as you know, on cryo-EM, and as I said, I'm pleased to see you. But I just wanted to very quickly show you that I've been working on that huge DNA PK molecule with 4,000 amino acids, and I couldn't see the drug molecules bound using X-rays. Using cryo-EM, we got all of the main molecules which are on the market are all defined like that. And the paper went straight into Nature again. You can see my objective in life is to get papers in Nature. But this is amazing because these drugs have been on the market for ages and nobody knew how they worked. But now using cryo-EM, you could see these small molecules in a molecule with 4,000 amino acids. So things are changing. and it's all very exciting. So what the other part of my group is doing is quite a lot of work, mutations, statistical methods, and machine learning, and that all fits in very nicely. So that's my story. I like to show some pictures of the group. These are the people who developed the cryo-EM as a facility, and I put them on the paper. And this is my team as of the last couple of years. And you'll see that we've got a reasonable balance of women. We've got a total international group, and there's several Indians there, and they're always useful, so always welcome. And I'll leave you with those thoughts. And we're just moving up to the new campus, but you can see what we've been doing. We're continuing to do some work on cystic fibrosis and on tuberculosis a little, but mainly on leprosy. Thank you very much.
It's time for a few questions. Anybody? Of course, there is a lot of time for interaction with Professor Dunn, over the next few hours. But if there is any questions, please.
You have to shout because I'm deaf, as you probably realize.
Very good morning. Very good morning, sir. This is Shruti here. It was an honor listening to you. I think it's a lifetime opportunity for me to listen to your stories. Sir, so I work with Syngene. So just that I'm into the cancer department. I work in R&D lab. So just to understand, there are a lot of data around. So how do you feel that artificial intelligence, your comments, that's it, that your comments on how do you feel artificial intelligence and all the soft data that we have is going to help us in fastening the drug discovery process?
Yeah. No, I think what you have to do is just be aware of the sort of things I was talking about in terms of molecular weight and all the other properties, and just do all the things you're doing in the academic lab you can do, and you can get things through first phase, into phase 1 in your academic lab. The challenge then is, can you get a company to come in and develop it? But I think you should try and do quite a lot of the work in your lab. I'm not sure whether that's what you're asking, but I think that's what you should do. Yeah.
Good morning, sir.
Good morning.
We are delighted to have you here today with us, and we really enjoyed your story and whatever work you did so far. So, sir, I want to know, has your group done anything in for targeting age-related brain disorders?
Sorry,
Because
Age-related brain disorder. Is there anything?
I haven't got into that area. Our company has a little bit, because the problem we've had in the company is I tried to keep it in cancer, but we actually bought 1 or 2 other companies which then got merged in, and some of them were working on those aspects. But I've personally not been very heavily involved, I'm sorry.
I see. Actually, sir, I've been reading a few articles since few days, which tells us about naturally available antibody targets to naturally available antibody agents used for targeting age-related brain disorders.
Yes.
So
Yes.
I wanted to know from you that how would you compare drug agents with antibody agents here?
Well, they're obviously both very effective. And I mean, as you can see, I've already spent my life being spread over all sorts of things. And I decided when I was thinking about the antibody side, I'd probably leave that to others. But yes, you can do a lot of work which is sensible and many people are. Yeah. So, go ahead.
Thank you, sir. Thank you.
Ah.
So, first and foremost, thank you for honoring us and honoring me with your wonderful lecture. I thought it was absolutely fascinating.
Thank
And
you.
I always enjoy a lesson in history when it comes to science. So, thank you so much for sharing all this wonderful history that you've gone through, which I think is going to be absolutely special for all of us. But really, the question I wanted to ask you, Sir Tom, is, you know, I was very intrigued by the way you've looked at evolution of, you know, various DNA.
Yes.
And I was wondering whether is that a way of also predicting certain cancer mutations, because after all, cancer is an evolutionary process. And is that a technique you can use?
Yes. So what I've done is to write a lot of software. Some of it's using statistical methods, and over the last 15 years or so, machine learning, AI. And so the problem with the protein is you get large numbers of mutations which are not going to have any impact. So what we've tended to do then is to go through various drug targets, look to see which mutations seem to be causing resistance, and also look to see, as I mentioned, which areas of the protein surface are less likely to be susceptible to change in order to get resistance built in. So we've done quite a lot of work on that, but it's still a big challenge. Yeah.
Thank you. Thank you.
But thank you for your kind words, by the way. And I'm looking at your background. I've seen a lot of things in parallel as we've done. So sort of
Well,
thinking
insulin
about
is something that, you know, really is something that you and I share in terms of our passion for.
Yeah. But I think the message for these people who in an earlier career is that you can actually contribute quite a lot if you think about what you're doing academically. You still need to make sure it's academically coherent and good. But I would keep an eye out. Yeah. Thank you.
Anybody else? Yeah, so if there are no further questions right now, obviously we will have a student interaction session with Professor Tom Blundell after the IT session. You can ask more questions at that time. With that, I request Kiran to hand over the moment to Professor Tom Blundell.
Goodness me. Amazing. Yeah. Thank you very much. Yes. Yes. Yeah, that's fantastic. Thank you. I have to consult how to get that back.
No, we'll send it.
Could you?
Just we
Yeah, okay, that would be helpful. Because I'm forever hiding things as I get on planes. Yeah.
Thank you so much, Professor Blundell. It was an honor. We were fortunate to hear this scintillating lecture and the history of science, as Kiran was saying, which is also equally very fascinating to me to hear about the journey that you took in reaching this level. So, just a couple of announcements. I request all of you to assemble at the lawn for the group photograph immediately after this session, which will be followed by a high tea after the photograph session. Please join us in both high tea as well as the photo session. Thank you.
Thank you. Yes, thank you. Sorry, I went on rather a long time. Was it all right? Sorry, I didn't put anything about you in. I should have done. I
so many colleagues.
well, I meant to, actually, I went home yesterday thinking I would try and find some time to add something in. I'm just going to pack this up.
The system.